Well, thank you very much for the introduction and for the invitation to speak. And we're focusing on the AIR community, which is a group that has developed to develop protocols for curating, for analyzing, and for, very importantly, sharing these massive data sets on antibody, B cell receptors, and T cell receptors in the AIR data commons. Next slide. And so the presentation, I'll introduce the AIR community and partly discuss why we need a new initiative for these type of data. And then Brian's going to talk about the AIR data commons and iReceptor. And then Kira will talk about some applications of all these data to diseases, including type 1 diabetes. So next, please. And so to introduce the AIR, the Adaptive Immune Receptor Repertoire community, it's really important to discuss why we need a new community initiative, as I say, for these type of data. And to do that, we have to introduce a little bit about the adaptive immune system and what is unique about it. And so this part will focus on the antibody B cell and T cell receptors sequences. We call that AIR-seq data. These massive new generation sequencing databases are very important to development of vaccines, autoimmune disease therapies, new cancer immunotherapies. One of the key parts of the adaptive immune system, why it's called the adaptive, is because it evolves within the individual as the individual develops a response to a pathogen. And for all of this, it has to be incredibly variable. And that's one reason why it's so difficult to deal with these data and why we really need a whole new community initiative. And, a lot of people are developing genomic resources, incorporating genomics and human health. But as I say, the unique aspects of the adaptive immune system have led us to start this initiative. And one of the main aspects of the adaptive immune system is its high diversity. It has to be incredibly variable to recognize all new pathogens that an individual may experience, or even newly evolving pathogens, i.e. the novel coronavirus. And partly these sequences are just incredibly diverse. And it's been estimated there's like 10 to the 13th potential B cell receptors in the human population. That doesn't mean any given individual would have 10 to the 13th, but you might have 10 to the 9th B cells at any time. They might not all be unique, but many of them are. And this produces huge data sets in that now samples can be anywhere from 10 to the 6th or 10 to the 7th sequences. And this might be before and after a vaccine trial, or within a tumor and outside a tumor. So again, the data sets are huge. And here's an example on the slide of systemic sclerosis, where just one study produced 700 million B cell receptor sequences. So every week we're getting these huge data sets. And one of the things that the AIR community was decided to take on was how can we curate these huge data sets in a usable way? So next slide. As I say, you have to learn a little bit or emphasize a little bit about the uniqueness of the adaptive immune system. And that slide shows partly where this diversity comes from. And it's from VDJ recombination. So when a B cell develops, there's multiple genes in the germline on the chromosomes of the immunoglobulin genes or the T cell receptor genes. And I like this slide because it actually shows a little metaphor of a scissors going in, and the initial variability of, let's say, a B cell clone or the differences between B cell clones is during its early development, it'll take one V gene, one of the many D genes, and one of the many J genes, and stitches them together by going in and cutting out the DNA between those V, D, and J genes. And so this is, in fact, unique in the eukaryote population. This is the only type of gene where where recombination at the chromosomal level occurs to produce this initial variability. And so clones are sets of these B cells and T cells descending from one of these initial VDJ recombinations. And of course, with B cells, then a B cell clone that binds to a new pathogen undergoes more diversity because it initiates somatic hypermutation, a mutational process. So you actually have a Darwinian process going on within the individual of selecting those clones that are most binding to the pathogen and then allowing them to mutate, and then another round of selection. So this is the one of the processes that leads to the huge diversity of B cell receptors or T cell receptor repertoires. And then, as I say, it's very unique. So it demands kind of special databases, special ways of analyzing the data, and the huge diversity of it demands special ways of handling the data. So next slide, please. And then really one of the other problems is that there's so many steps in going from a sample to a final representation of an individual's B cell or T cell receptor repertoires, and the experiments can vary slightly at any of those steps. And then the next slide. So we're trying to say why these data sets are so complicated and need new ways of analyzing and curating them. And this just shows the importance of them. When we talk about clonal expansion, on the right side, this is this is a study of chronic lymphocytic leukemia, or CLL. On the right side, you have a healthy donor. These little gray blobs are clones of B cells in the healthy donor, and they're pretty much distributed. While on the left side, you see a few clones that have completely overgrown in the blood, in the PBMCs. And so this just shows how clearly this clonal expansion is kind of one of the keys to understanding and treating and following these diseases that involve B cell receptors and T cell receptors. So next slide, please. So that was kind of a review of why we need this. The AIR community got together in 2015, where immunologists and computer scientists and experts in ethics and data storage. And we developed protocols for curation and storage and analysis of these data sets. Next slide, please. And the key to the AIR community really is the working groups. We have working groups that take on particular aspects of this greater initiative. We have a germline database, legal and ethics, and then biological resources, common repository. And then next slide, please. And again, one of the keys is we're really trying to get community buy-in. And so one way we do that is that all of these protocols that we are putting out there to the community, they're published in papers. And the first thing they do is they get accepted and reviewed by the entire AIR community. So that's one way we try to say that this is a community initiative and anybody can join these working groups. And in fact, we hope everybody who's interested would join these working groups. And on the slide here, we have one way to start working with the community is air-community.org. As I say, these working groups are open to all individuals interested, and we greatly encourage people to come and work with us on developing these community standards for curating, sharing, and analyzing these data. So now I'm going to pass it on to Brian to talk about more of the guts of the AIR Data Commons and iReceptor. Thank you.
Thanks, Felix. Yeah, so I'm going to talk a little bit more detail about what the AIR Data Commons is, and then I'll dive into a little bit about iReceptor and how it works. And then Kira is going to give you some detailed overviews of how to use iReceptor if you're interested. So Felix mentioned this already, and I'll just kind of reiterate a little bit. So the reason to have an AIR Data Commons is that we want to be able to select data across many studies, many different research groups. All of these data sets are really, really large. They're talking about billions of sequences and hopefully scaling up to tens and hundreds of repositories in the future. And one of the things about having a distributed AIR Data Commons, so repositories distributed around the world, is that this allows researchers and research groups to curate and manage their own data at their home institution under local data policy if they so choose. And one of the goals of the AIR Data Commons is really to make AIR-seq data that is fair. So FAIR, if you're not familiar with it, stands for findable, accessible, interoperable, and reusable. So really what we want to be able to do is make it so that researchers can find this type of data easily. You want it to be able to access that, and interoperability is really, really important. So you basically want to be able to compare data sets across studies. And ultimately, the goal is to find and reuse data. And the main goal for that is, of course, to derive new scientific insights from the data. So the iReceptor approach is basically kind of twofold. We have the iReceptor Gateway, which is basically a web portal. And Kira is going to talk about this in a lot more detail later, so I won't go into too much detail. But it's basically an interactive web-based data discovery platform that allows you to do data exploration and analytics of the data. But what it's actually doing that analytics and exploration of is data across the entire AIR Data Commons. And the AIR Data Commons is, as I said, a distributed set of repositories that are distributed around the world. And the reason this works is because of the standards that are set out by the AIR community that Felix was talking about. So the only reason you can actually query and interoperate across data across these repositories is because that they're standards based. And so what the gateway does basically is sends out queries to all these repositories. So if you're looking for a specific disease, a specific type of subject that you're interested in, you can send queries basically from the gateway. So you're typing at the gateway, you kind of type in a disease. I'm interested in type 1 diabetes data. It sends data queries across these repositories around the world, and it brings back data. And so really what the gateway is doing is hiding the complexity from the user that you're actually searching a bunch of data around the world. So the AIR Data Commons has been around for a while. As Felix mentioned, this initiative started in 2015. And in 2018, we had on the order of kind of 500 million annotated sequences across maybe a small number of 10 studies, 12 studies, or something like that. But the AIR Data Commons has grown pretty dramatically over the last 5 years to almost, it's approaching 6 billion. Right now it's about 5.2 million, but we anticipate it's going to be approaching 6 billion rearrangements, annotated sequences. So these are sequences that are annotated with VDJ and CDR3. So you know the VD, DG, and JG and CDR3. So these are not just raw sequences. They're highly valuable annotated sequences. So they've been run through those processing pipelines that Felix talked about. And the data repositories have grown. Sometimes they grow in small step increases when you add a study into a repository. One of our partners, VDJ Server from the University of Texas Southwestern Medical School, they tend to add data in very large step increases. So they add tens of studies at a time. And so that's what these big steps are. And one of the interesting things that I'll talk about a bit more is the impact of COVID-19. So when COVID-19 hit, all people got very, very interested in B-cell and T-cell data sets that involve COVID-19. And we had a big spike of growth there. And I think one of the really exciting things from my perspective is that we actually now have, in those early days, kind of up to 2021, we had kind of a small number of large repositories. Over the last year, we basically added 5 new international repositories, and we're adding more and more repositories all the time. So we're scaling up in terms of the number of repositories, and those repositories are distributed around the world, as you can see. And interestingly enough, from a type 1 diabetes perspective, we have so we're working with a group of type 1 diabetes researchers, and we have a new type 1 diabetes repository that came on just this month. In terms of usage trends for the gateway and the iReceptor platform, we have a very large number of users. This is the number of users added every month over the last couple of years. And so we add on the order of 10 to 15 new users a month on average over the last year or two. We had a very, very large interest in the platform, spike in interest in the platform in June of 2020 when we had new COVID-19 data. One of the interesting things to point out, it's a very international resource. So our users are spread around the world. Basically every country, every continent. And one of the interesting things is we have a fair number of industry users. So 24% of the new user accounts in the last year came from people in industry. So I'm going to talk briefly about COVID-19 and how we've kind of how we've seen that driving use of the AIR Data Commons. So one of the things that we were able to do, because we were kind of in place and ready, up and running when COVID-19 hit, the first studies, a couple studies, the first study was from Nielsen, from Scott Boyd's group. And their data came online, was available in, I think, probably April or May. We had that data up and in the AIR Data Commons annotated by June. So we were able to act very, very quickly. And over the next several months, a number of other studies were published, and we were able to add those to the AIR Data Commons. And one of the key things that really triggered that spike in interest is that we worked with Schulthess et al. And the Binder group to basically publish this data from this study. It's a very, very rich study. It had 46 subjects. It had both IG and TR data, 15 million annotations. And it was one of the first COVID-19 time series studies available. So you could look at trajectory over time of disease progression of COVID-19. And some of the subjects had data points up to 9 time points in the study. So this is a very exciting study, and that's what really drove that spike in use. So these studies progressed over time, and currently we have, so COVID-19 data, there are over 1 billion COVID-19 annotated sequences in the AIR Data Commons. It is our largest single disease type. And this is a really, really good example of kind of disease-specific data sharing driving research. Now, this part of the puzzle is about kind of publishing data in the AIR Data Commons, but really the key thing about the AIR Data Commons is being able to reuse the data. And so this is just a set of papers. I'm not going to talk about any of them in detail, but these are all papers that cited the iReceptor platform on the AIR Data Commons for reusing COVID-19 data. So these researchers are not publishing data into the AIR Data Commons. These researchers are taking data out of the AIR Data Commons to support their research, right? And that's really what it's all about. I'm going to give a brief overview of iReceptor version 4.0, which we just released in December. And then Kira will take it from there to dive into some of the details. So historically, kind of the AIR Data Commons and the iReceptor platform was about bulk AIR-seq data. So these tens of millions of sequences per study. So it's very, very deep sampling across many, many cells. But you typically only get single chain annotations. So it's very deep sampling, but you don't have a really strong understanding of kind of the behavior of the immune system. With single cell immune profiling, so I think most of us are probably aware of single cell immune profiling and how that's kind of become very, very prominent over the last 3 or 4 years. And so single cell immune profiling gives you paired chains. So you kind of have a heavy and light chain. So you have a much better understanding of the kind of the binding of a specific cell. You have much higher resolution in clones, and you get gene expression data. So you have a much better understanding of kind of the phenotype of what the cell is and how what the state of that cell is. So bulk AIR-seq and single cell scRNA-seq data is actually quite complementary. And so one of the things that's happened is we've been working on integrating this data, and I'll tell a bit about that in a minute. So one of the things that you get typically with AIR-seq data without single cell data is you kind of can identify clonal lineages and kind of highly expressed clones in individual. And that might be interesting because of disease progression and trying to understand how the immune system is fighting that disease. And so what you get is that single clone, but with gene expression and single cell data, what you're able to do is get a very much better understanding of how that cell fits into the kind of landscape of the immune system and how the immune system is functioning. So with iReceptor version 4.0, we're able to curate both AIR-seq data and single cell scRNA-seq and gene expression data in the AIR Data Commons. And so the reason that's happened is, again, primarily because of the AIR community. So the AIR community has been spending the last 2 to 3 years working on a single cell and gene expression extension to the AIR standards. And so that is currently available. It was released in August of 2022. And that allows you to basically load gene expression and cell data into AIR Data Commons repositories. If you're familiar with kind of the 10x single cell pipeline, it's basically loading the matrix features and barcodes files, if that is familiar to people. And so what you're able to do with the gateway is query studies based on both VDJ and cell and gene expression data. So as I mentioned, iReceptor version 4.0 came into production in December of last year. And we have a repository software stack, so you can store and load query and query single cell and gene expression data. And the iReceptor gateway has some single cell immunology workflows that Kira is going to talk about in a minute, actually right now. Oh, no, sorry, I have one more slide. Yeah, so kind of one of the things that's changed in iReceptor 4.0 is we've gone from kind of being a user interface to the AIR Data Commons to a platform that's trying to integrate data from a bunch of different platforms and a bunch of different modalities. So we have bulk AIR-seq data, we have single cell scRNA-seq data. And so we've gone from becoming a platform for finding and downloading data to more a platform that can kind of perform a much richer set of analyses. So we have clones, cells, and gene expression data through the AIR version 1.4 standard extension. We have clone, cell, and gene expression analysis workflows on the gateway. We spend a lot of time in the AIR community making sure data is interoperable by using ontologies across the board for many of the standards fields. So when you're looking for a disease, you're looking for an ontology ID for a disease, not just an arbitrary name. So that makes data much more discoverable and interoperable. We're linking out to other repositories. So we talk to, if people are familiar with IEDB, it's the International Epitope Database, and it contains information about antigen specificity. So if you search for a CDR3 on the gateway, on the iReceptor gateway, that query will also query IEDB, and it'll find out if it has any known known antigen specificity for that CDR3. And we're looking at other things like integrating and connecting to Human Cell Atlas and other types of platforms as well. And another big thing with the gateway that we've released in version 4.0 is, so as I said previously, you could find data and download it and do analytics on your own. We now have an integrated analytics capability in the platform. So we integrate complex analysis tools into the platform, so you can download you can find data, but you can also analyze it and do some very basic types of analyses within the platform. So you don't have to install analysis tools on your desktop or on your local computer. You can do things just within the gateway. And with that, I will stop sharing because Kira is going to take over.
All right. Getting set up.
And I will try to answer some of the questions online while Kira is talking.
Great. So I'm going to be in this last part of the talk, just providing sort of how to navigate the iReceptor platform, specifically taking into account some examples. No, oops, gotta get that. Taking into account some examples from the T1D literature, and just going into a bit more detail about how to navigate these new 4.0 features that Brian described. So you've heard a lot about the gateway, but what does it look like and how does it work? So as Brian described, the gateway searches the AIR Data Commons. Presently, this is 8 repositories, 86 studies, over 9,300 repertoires, and over 5 billion sequence annotations. So when you first log into the gateway, you see a screen that looks like this, and it shows us the pie charts there are showing a snapshot of the available data. So we have multiple different study types. Most of the data is from humans, a little bit from mouse. We have 57 diagnoses, as well as a bunch of healthy control data, 43 tissues, all the immune receptor loci and different target substrates. And there's two key workflows that we've integrated into the gateway. So these are searching study metadata and searching sequence annotations. And you can activate these workflows from the home page like this. So this button here, you can browse all the repertoire metadata, and later I'll be talking about this sequence quick search, where you can search the AIR data commons for any CDR3 of interest. So how can we find data in the AIR data commons using the iReceptor gateway? So when you click on that browse repertoire metadata button, you get a screen that allows you to view all the repertoires and their metadata that are available. You can, of course, filter this data using either a full text search keyword, and you can filter by multiple study, subject, and sample filters. And this is sort of the default view that's provided. Each field is a AIR standard term. However, you can customize the displayed columns. If you click the button, then you can have a pop-up where you can actually display any of these over 80 study, subject, and sample AIR standard metadata fields, should they be curated with the data. And a new one that we recently added or now have available due to recent addition of the standards is these MHC genotype fields. So this is a new capability that we've integrated. And so if the data is HLA typed, then you can do a keyword for a specific allele of interest. And I'll note that the recent T1D data from our collaboration, so this is all subjects from the DAISY cohort, it's fully HLA typed. So as I mentioned, the AIR data commons has a bunch of healthy control data. So to access this data, you can apply a filter. So here I've applied a filter for the study group. Just looking for healthy controls. And this filters down all the data to about 260 million sequences from 222 repertoires. And this is from 12 different studies. And we also provide sort of high level repertoire statistics for each repertoire if you click on the stats button. So here I'm showing a pop-up where you can see the V gene, D gene, and J gene usage distribution, as well as the junction length distribution. And these are interactive statistics. So here I'm showing the V gene distribution at the family level. But if you click on any of these bars, it'll sort of drill you down into the next level. So here I'm showing gene, and we can also take it further to the allele level. So getting back to our healthy control data, once you've filtered down to your repertoires of interest, you can then browse the sequences. So this opens up the second phase of the search, so the sequence search. So what we have presented to you in the gateway is the V gene, the D gene, the J gene, and the junction CDR3 amino acid sequence, as well as the junction length. And you can filter down these sequences based off of the VDJ genes and the junction AA length. And once you've found sequences that you're happy with and they you can sort of peruse that interactively, they look good, then you can download these for offline analysis. So the next few slides, I'm using sort of some examples from the T1D literature to showcase some of the cool features in the gateway. So first feature being the sequence quick search. So for this, I pulled up a paper from T1D literature. This paper is called CD8 T cells specific for the islet autoantigen IGRP are restricted in their T cell receptor chain usage. So what they found here was that there was a certain motif, which I've highlighted in yellow there, that the motif corresponds to TRA J53. was enriched in clone types within this cell population. So this is a perfect example to illustrate the quick search. Basically, I can take that sequence, pop it into the quick search box, and search all of the data in the AIR Commons for this sequence. And the result is that there is over 2.7 million sequences found from 3,585 repertoires. And this is just showing a fraction of the data, but it's the J gene is TRAJ53, as we would expect. And interestingly, this sequence is found across over 2,400 subjects. So 2,400 different subjects. So it's a very public sequence. The data is all from the TRA locus, as you would expect. And also interesting is the fact that this islet-specific TCR is present in subjects who have 26 different diagnoses. So none of these actually are T1D data, because presently we don't have any TRA data within the AIR Data Commons. The DAISY study actually right now is all TRB. So this is all from non-T1D individuals, and there's actually a fair amount of the diagnosis, which is called none, which basically means they're healthy. So this, to me, as a non-expert in the type 1 diabetes field, was quite interesting. But it actually does agree with some previous work. So here I'm showing a recent review using the T-cell receptor as a biomarker in type 1 diabetes. They say that it's actually still controversial whether T1D patients have distinct islet antigen-specific T-cell subsets in the blood compared to healthy people. And a number of studies actually do indicate that healthy people do have islet antigen-specific T cells. So what we can say from here is that just with a few, quick clicks on the gateway, you can find some interesting results. So it's a very powerful system. So for the next sort of use case, I want to describe our antigen specificity feature, which is an integration with IEDB. So that's the Immune Epitope database. And so again, using the same review paper from before, I pulled this highlighted this part here where they talk about these non-diabetic NOD mice, which is an animal model for T1D, and they spontaneously develop diabetes, and this is associated with insulin autoantibodies. And these many studies have been able to identify CD4 T cell clones in this mouse model that are reactive to insulin. So this my example here is let's use the gateway to identify some insulin binding TCRs. And first for that, I went to IEDB. I did a search to identify insulin binding TCRs. So my filters here are the antigen being insulin, the disease being type 1 diabetes. This results in 206 receptors. These are all TCRs. And we can take one of these, so just taking the first one in the list here, we can take one of these chains. This is the beta chain. Recall that the T1D data that we have presently is all from TRB. So we can take this chain, pop it into the sequence quick search, and search the gateway, or search the AIR Data Commons using the gateway. And the result is that there's 5 sequences of this specific TRB, CDR3, and it's from 4 different repertoires or 4 different samples, and they're all these are all type 1 diabetes subjects. And this was sort of getting to what Brian was mentioning before about our feature where we integrate the CDR3 search with the IEDB, is that when a user activates a CDR3 search, the gateway will simultaneously query IEDB for any known binding interactions. And as you see on the left there, it provides a pop-up of information letting the user know if there is any of these curated interactions. So in this case, because I searching for TCRs known to be specific for the human insulin protein, we would expect to see that pop up as some known specificity. But interestingly enough, there was also some bacterial antigens that popped up. So one hypothesis in the T1D field is that these TCRs could be associated with previous infection. So this feature of the gateway really allows the user to discover some previously unknown to them specificity that they weren't aware of. So it really makes the gateway even more powerful for doing some pretty advanced science. So just to show that this was sort of not a quirk of the gateway, we can reproduce the findings in IEDB. So here I'm basically just doing a reverse search, searching IEDB for that same chain. And you see these antigens popping up for the 3 different organisms. So insulin, as you would expect, and then these 2 bacterial proteins. So now I'm just going to take some time to go through in more detail some of the new and exciting features for iReceptor version 4.0. So these are clone, cell, and analysis capabilities. So whereas in the previous workflow, I presented the sequence level workflow, we now have workflows for finding clone and cell data. So this is what I'm showing here is a filter on the data to show a single cell study. This is a 10x immune profiling study, which has sequences, clones, and cell data, so gene expression data. So looking at the clone UI, so when you go to browse the clones, this brings you to the next phase of the search. It looks pretty similar to the rearrangement sequence level view. You have the V, D, and J calls. You have the junction AA sequence, but now you also have the tool clone ID, the clone counts, so how many members of the clone, and then the abundance of the clone. And like before, you can apply different filters. And for the cell page, to look at the cells, you can see that, so in this view, each row is one cell, and we're showing the 2 different chains, so chain 1, chain 2, and showing also the V gene and the CDR3 for each of these chains. So it's nice presentation for the user. Everything's sort of right there at your fingertips. And we also show the top 4 genes expressed in each cell. So based off of their gene expression, this is the RNA-seq expression. And you can filter down this data by different features. And most sort of interesting one is to apply a cell expression based filter. So you can do a filter on any gene, and you can you can define a sort of minimum expression value that you're interested in for that gene, and it allows you to filter down the data. And finally, I'll just briefly touch on some of the analysis applications. So whereas in iReceptor version 3.0, the whole deal was that you could sort of select your data of interest, but you would have to do your analyses offline. And now in 4.0, you can actually run analyses on the fly on the data that you find. So the user and the data never leave the platform. So here I'm just showing some of the user interface for the analysis app. So we have a handful of apps integrated into the platform. These are a mix of community developed open source apps, as well as some that we've developed internally. Here I'm showing Cell Typist, which is a single cell annotation tool. So the user can submit an analysis job on their data, and the gateway does all the work. So it downloads the data from the Air Data Commons, it stages it to computation, it gets the app ready for computation, runs the job, tabulates the results, and presents the results to the user in a nice digestible way. So here's the job page for our cell typist job. In this case, we ran cell typist on 5 different repertoires, and the gateway provides all the analysis available for download if you go to that analysis details tab, but it also presents to the user a summary. So if you click on the view summary button, you can view an HTML page where it just captures some of the sort of high level key figures. So in this case, I'm showing the UMAP from cell typist. So there's cell classification per repertoire. And these are just some of the example output available for apps. So here, apps at the sequence and clone level, you can get VJ gene usage heat maps. We have a histogram app that allows you to construct a histogram for any of the relevant AIR fields. We can look at the top sequences and look at the abundance of sequences in different sort of visualization ways. And for cells, we have an app called Conga, which simultaneously clusters T cells based off of their gene expression as well as their TCR sequence. So getting more sort of fine grained level of the clustering. And you saw cell typist before. So at this point, I think I'm going to pass it over to Felix for a quick wrap up.
Great. Yeah, well, thanks to both of you for, and just as a wrap up, just want to highlight, we mentioned that we were expanding in collaboration with several type 1 diabetes research groups. But really, as a general statement also, we're hoping to achieve what you might call a network effect, where the AIR Data Commons becomes that kind of go-to place to put your data in a very usable form, to emphasize that AIR Data Commons and iReceptor doesn't do anything anybody couldn't do, but just facilitates it and makes it 100 times easier to get at these data in a usable form, in a well-curated form. We had a lot of success with COVID-19 sharing and community support, and we're hoping to expand that to other diseases, including type 1 diabetes. And then it's a lot of people are understanding that the AIR-seq data, these immune receptor data, don't really live alone. And again, pushing the single cell work, you're then bringing in gene expression and extra data on cell phenotype. And so a lot of the initiatives at the community level, the AIR community level, and the iReceptor database, scientific gateway level, are focusing on combining the single cell data with the bulk sequencing. So kind of with the AIR Data Commons, if you want to get start using the iReceptor or get any questions answered about the AIR Data Commons and using the AIR Data Commons, please write to support@ireceptor.org. And then we have some of our colleagues and funding sources in the next slide, but I think we can open it to questions then and send it back to Corrie.
Okay, so now is our Q&A session, and thanks for the great presentation. So for all the audience, if you have questions, you can post in the chat or just unmute yourself and join the discussion. So I know there's several questions during the presentation already, and some of them already answered, but I will go into repeat it first. So people who are taking the on-demand webinar or watch the recording, they will know. There's some question is sending to me private, so I'm going to ask the question. It's a very general question. So first one is, when I visit the iReceptor gateway, it seems like I have to log in. Do I need to join as a member or do the collaboration first to get access to all the sequence?
Yeah, you just need to send an email to support@ireceptor.org to ask for an account. It's completely open. We do manage things through accounts so that we can track usage. Some of our funding agencies require that. So, but it's completely open. And the AIR Data Commons, so one of the things, so it's important to kind of split up what the AIR Data Commons is and what iReceptor Gateway is. So the AIR Data Commons is a set of repositories. There's, I think, 18 of them in total that use this query API and they're completely open, and people can do things with them. They can send queries directly from their own software if they want. What the gateway does is make that really, really easy for people to do. So the AIR Commons repositories are completely open. The iReceptor gateway has an account system, but you can just send an email to support@ireceptor.org, and we'll give you an account.
Okay, thank you, Brian. And then there is second question. Let me look in. So if I want to contribute the data, do you accept data other than human being, like other organism, or it's just a repository that accept human data?
No, we have some mouse studies in the repository, so it's not human specific at all. 99% of our data is from human subjects, but that is not a restriction. One of the challenges we have is these data sets are quite large and quite hard to manage. One of the repositories, VDJ server, accepts user submissions. And we have a software stack that research groups can use to store their own data locally in an AIR repository that's AIR compliant. That's completely open source and freely available. If anybody's interested in that, they can reach out to us. And so those are kind of the two mechanisms to share data of your own. So.
Okay, thank you. And then I think there's some question from Anita, and that also already been answered, but I'm going to just repeat the question so people will see it, because they won't be able to see chat during the recording. So what is the difference or relationship between AIR and the ImmuneDB?
Yeah, so ImmuneDB is a repository that stores AIR-seq data. And it actually stores it according to, and you can extract data from as AIR standard compliant data. What it doesn't do is it doesn't implement the query API yet. We've actually been working with that group to actually have that become an AIR compliant queryable repository. So it's not part of the AIR Data Commons, but it stores AIR standard compliant data. I don't know if there's a subtle difference there. I hope that answers that question.
And I guess we've emphasized enough that, I mean, there is a turnkey that if people want to set up their own repository, right, and they can get that, and we can help them with the technical part of it, and then they can start, exposing their own data to the AIR Data Commons. And I think partly with Anita's questions, I mean, we've been working, the AIR community has been working, since 2015, and we've made a lot of progress, but the more progress we could make in developing the AIR Data Commons, the better. And part of it, I think, is somehow having a way to get researchers who put the data in the AIR Data Commons get kind of an immediate feedback onto who's downloading the data, who's using their data. We're trying to work on that.
Mm-hmm. Okay, and I think Anita is not here, but there's a few questions. How do you track usage of AIR? I think it's human data in the scientific literature or resource itself. And then do you know there's RID for the resource? And then, yeah, I think that's the first question from.
Yeah, so all of that kind of tracking that I showed with all of those papers, those are papers that are actually cited, the iReceptor paper or the AIR Data Commons paper. So those are basically paper citations. And we encourage people to both cite the AIR Data Commons if they get data from the AIR Data Commons, and also cite the originating paper that produced the data, right? That's really important. You have to kind of acknowledge the producers of the data as well as the platform where you got the data from. And I know about RIDs. I was a little bit surprised that we do have RIDs for the resource, and I'm not sure how those were minted. I think so. All of the AIR resources are available on fair sharing, so I'm guessing that probably RIDs were produced by looking at the fairsharing.org site, I guess, but I don't know.
I think when there are new resources, if we get the information, we usually try to add it to the registry and then advertise it to other community to let them know this resource. So maybe, yeah, if there's new paper published, but resource owner itself can contact the registry and register the data, the resource. Yeah, and then I think most journal right now, like people citing RID in the journal, so they can track the usage of the resource. Yeah. So there's one question is, do you also query commercial data about these epitopes from antibodyregistry.org?
So we don't at the moment. I think that's one of the interesting directions where things are going, is there's lots of different, like I said, in our first version of iReceptor, or iReceptor version 3, so it's not our first version, it's version 3. It was still targeted at just querying the AIR data commons, whereas iReceptor version 4 is very much reaching out into querying other repositories that have related data. And so we're kind of exploring where to go next. There are lots of different options with those things. And so, but currently really the only resource that we're querying outside is IEDB at the moment. And then we use ontology, kind of EBI's OLS ontology query mechanism for our managing our ontologies. So those are the two external things that we're querying at the moment.
Okay. And thank you. And I think because Anita not here, but there is some discussion about the identifier. Brian, do you want to address that part? I think you mentioned you use the bioproject and biosample for some identifier.
Yeah. So, I mean, again, it's just make it really clear. So all of the data in the AIR data commons is essentially processed, and we consider quite value added data. So basically it takes data from NCBI repositories like SRA, raw sequence data of B cell and T cell data, and then it's run through those annotation pipelines that Felix mentioned early on in the talk. And those pipelines are very, very complex. And so if you had to run those, if every researcher who wanted to reuse data from SRA and wanted to run it through that pipeline, that would be an enormous amount of additional work every time somebody wanted to reuse the data. So we have the process data. We consider it quite value added process data. And it's that data that you're using when you're querying the AIR data commons. We store within the repositories bioproject, and quite often sample or run identifiers from SRA and NCBI so that researchers can link back to the original data if they want to. So we're trying to use PIDs, the persistent identifiers that exist in the NCBI repositories within our repository, so you can cross-link the data. And then when I was looking at the SciCrunch site, and I noticed that they have, so there are SRRs minted or RIDs minted, for biosamples. So there's a kind of an RRID PID for each biosample, which we don't link to. We link to the biosample directly. So that's kind of where we're at the moment. We definitely cross-link back to the PIDs in the NCBI repositories as much as possible, so that researchers can find that original data. And if you look at the gateway, when there's a project ID in the gateway, it's typically a PRJ-NA, so it's a bioproject project ID. And if you click on it, it'll actually take you to the NCBI site, so you can look at the original data directly from the gateway.
Okay, thank you. So any other people, you could unmute yourself if you would like to join the discussion. And then if not, I saw there's a question. So from the audience, let me find this. Okay. So in the abstract, there's a list of top 3 most question. I think the audience want to know about the second one. Can you explain, can machine learning be used to identify individuals who will respond well to a new cancer immunotherapy based on difference in their antibody, B cell, T cell receptor repertoire as curated in the AIR data commons?
Well,
That's a great question.
It's a great question. I mean, just kind of in summary, you know, a lot of that's really some of the questions that a lot of these AI approaches are trying to do, and, you know, look at these deep sequencing, highly diverse repertoires of either B cell or T cell receptor repertoires and look for these signals. And, you know, I think the important point there really is that these AI approaches need huge data sets for both the training and then to do the analysis. And one of the places to come to get those data, those huge data sets and multiple data sets, are in the AIR Data Commons. So that's one reason I put it in as one of the, you know, I think a primary use of the AIR Data Commons in right now and in the near future is to look at these approaches. And again, you have to look across multiple data sets, and that's really what's provided by the AIR Data Commons. So I think that's going to be a more and more important use. You know, we've got a whole working group in the AIR Data Commons called Diagnostics and Therapeutics, kind of trying to bring those people together and talk about how to actually use these data to develop, you know, real screening and patient partitioning. You know, so that's a big thing that people are trying to do.
Okay, thank you. And I think the other two questions, probably we already do some use case in the presentation, right? Or do you want to add more comment on the other two questions in case they have question later during the on-demand?
You know, I think, you know, again, it's just type of uses that you can use for these, you know, these type of AIR-seq data. And then it's just really important. Typically, you know, a lot of even though they might have 500 million sequences, they only have 20 patients and 20 controls. So it really, it's one of these use cases for something like the AIR Data Commons, where you've got huge amounts of data, but not huge number of samples. And so you need to look at other studies, maybe even other diseases for comparisons. And that's what we're trying to do with the AIR Data Commons and the iReceptor Gateway.